Good morning, students. Today I'm going to move forward with virulence factors, in which our last virulence factor, which are toxins, and I'm also going to cover the natural defense mechanisms. Okay, so starting with the toxins. What are toxins basically? Toxins are basically the poisonous substances that are produced by certain microorganisms. Okay, there are poisonous substances that are produced by several microorganisms. Okay, and a toxin may be as simple as an ion or an atom which basically negatively interferes with a cell. Okay, and these toxins are basically the, the substances which harm our body, and the effects of the toxin can be determined with the biochemical reactions which takes place when a potential toxin is introduced to an organism. Okay, as well as the toxicologist. Okay, the persons who study toxins, okay, need also to study the effect of environment, pressure, heat, okay, as well as the property of the organisms which basically harm the human beings. Okay, and the property of the toxin is needed to be studied. Okay, and the toxins are divided into two groups, of which one of them is exotoxin, another one it's an endotoxin. Exotoxin and endotoxins. Okay, so what is the basic difference? Okay, what is the basic difference between exotoxins and endotoxins? What are the basic differences of an endo and exotoxins? Okay, before moving for that one. Okay, and how are basically toxins transported? Okay, and what are the effects? Okay, and toxins are basically the primary factor contributing to the pathogenic properties of the microbes, and the capability of the microorganism. If you want to note down, please note down the capacity of the organism to produce toxins is called toxigenicity. Okay, I am repeating the definition again of the toxigenicity. The capacity of the organisms to produce toxins is called toxigenicity. Okay, and the toxins are transported by the blood or lymph, which can cause serious or sometimes fatal effects in the human body also. Okay, and some toxins can produce fever, cardiovascular disturbances, diarrhea, and even the shock. Okay, depending on the type of toxins. And toxins also inhibit the protein synthesis, sometimes destroys the blood cells, even the vessels, and disrupts the nervous system by causing spasms. Okay, by causing spasms. What are spasms? Okay, what are spasms? Muscle spasms. Okay, simple a word, spasms, spasm. Okay, it's just an sudden involuntary contraction of a muscle and a group of muscles or a hollow organ such as heart. Okay, and it may be caused due to disease or various other factors also. Okay, due to which these spasms occur. Okay, and what happens actually in the spasms? Okay, the cramping occurs. Okay, a cramping of leg and muscles, the cramping of leg, okay, leg muscles or whatever the place where cramping occurs. Okay, and cramping may also occur due to another problems also. Okay, there are also other problems due to which cramping also occurs, but it is specific or it depends on the type of problem a person is having. Okay. So the spasms, okay, and of around by today or almost a year, sorry, till now about 220 or so known bacterial toxins nearly cause 40% of 40% cause disease by damaging eukaryotic cell membranes. Okay, then comes a term toxemia. Okay, then comes a term toxemia. What is toxemia? It is just refers to the presence of toxins in the blood. Okay, please write down, the toxemia refers to the presence of toxins in the blood. Okay. Now there are also another word which you have already studied in your semester 4, intoxications. Okay, intoxications. What is intoxication? Okay, it is due to the toxins, okay, presence of toxins in the human body, but the microbial growth is not necessary. But the microbial growth is not necessary. Okay. Now, what are exotoxins? What are endotoxins? Basically, endotoxins are produced inside, whereas exotoxins are produced inside some bacteria as a part of their growth and metabolism, but are secreted by the bacterium into the surrounding medium. Okay, simply endo refers inside, and exo in the toxin refers to the outside. Okay, so the exotoxins are basically proteins produced inside the pathogenic bacteria, most common in the case of gram positive bacteria, as a part of their growth and metabolism. Okay, and exotoxins are then secreted into the surrounding medium during the log phase. Okay, now comes the, okay, the photograph given here. Okay, it's a just diagram where you can see exotoxins are released outside the cell. And another example which is able to produce a exotoxin is a Clostridium botulinum. Now comes the endotoxins. What are they? They are lipid portions. They are lipid portions of lipopolysaccharides that are part of the outer membrane of the cell wall of the gram negative bacteria. Okay, so I am again repeating it. They are the lipid portions. Okay, they are the lipid portions of the lipopolysaccharide that are part of the outer membrane of the cell wall of gram negative bacteria, basically also known as lipidae, and the endotoxins are liberated when the bacteria die or the cell wall lyses or the bacteria breaks apart Okay, and you can see here endotoxins, okay, are composed basically of lipids which are basically parts of the cell wall. Now here it's a diagram here where you can see if we detach them, it will have a similar this type of look. Okay, one example is given here is of a Salmonella typhimurium, okay, which is a gram negative bacteria which produces endotoxins. Now, what are the differences between endo and exotoxins? Okay, everyone, please remember this table which I am going to discuss. It's very important. Okay, for exam point of view. Okay, for the exam point of view. Okay, and in many of the past papers, this question has been asked frequently in the exams. Okay, what are the differences between the exo and endotoxins? Okay, or write a note on toxins. It may be 5 marks question also. Okay, you need to remember this table which contains all the information about the exo and endotoxins. Okay, so starting with the definitions of both endo and exotoxins. Okay, first starting with the definitions. In the case of exotoxins, proteins produced inside the pathogenic bacteria as a part of their growth and metabolism, Whereas in the case of endotoxins, lipid portions of the lipopolysaccharides, okay, that are part of the outer membrane of the bacteria. But in this case, exotoxins, these toxins are basically released. These toxins are basically released. Okay. Now, how are they produced and produced by which type of bacteria? Okay, it's just for the generalized information. It is produced by most probably gram positive bacteria, but also some are also produced by the gram negative bacteria as well. Endotoxins, in most cases, gram negative bacteria produce endotoxins. Now comes the chemical nature of the toxins. Okay, these are protein complexes which are of polypeptides, okay, whereas the endotoxins are lipopolysaccharide protein complexes. Lipopolysaccharide protein complexes. Now comes the molecular weight of the toxins. For exotoxin, it is about 10 kilodaltons, and it is just an approximate weight, okay, whereas endotoxins range between 50 to 1000 kilodaltons, 50 to 1000 kilodaltons, okay. Now, which are the components of the exo and endotoxins? Usually composed of two subunits A and B, and the subunit A is seen to have a catalytic activity, whereas subunit B is required for binding with a appropriate cell receptor. Okay, I am going to discuss this later on in later part of this lecture. Okay, how this subunit are basically, or what is the basic mechanism of these subunits A and B. Whereas endotoxins are basically made up of three basic components: O antigen, okay, O antigen, a core oligosaccharide, and a lipid A, okay, and a lipid A. Now, presence of enzyme in the toxins, okay, or the enzymatic activity, you can say more more specifically, okay, which type of enzymatic activity this may have. It may be hyaluronidase, collagenase, certain proteases, nucleases, neuraminidase, certain proteases, as well as phospholipase A, okay, as well as for phospholipase A, okay. Whereas in the case of endotoxins, it may be catalase, it may be fibrinolysin, or it may be IgA or IgG proteases, okay. I'm again repeating it. The enzymes which may be enzymatic activity, more specifically, which may be present in the toxins. In the case of exotoxins, it includes hyaluronidase. Next comes collagenase, certain protease, nuclease, uraminidase, certain proteases, okay, and phospholipase A. And in the case of endotoxins, it is catalase, fibrinolysin, IgA or IgG proteases in the case of endotoxins. Now moving forward with the chromosomal locations. In the case of exotoxins, okay, it is located on the extra chromosomal genes. It is located in the on the extra chromosomal genes. Examples are plasmids. Examples are plasmids. Well, but where are the endotoxins located? Are located on the chromosomal genes, but not the extra chromosomal ones. Okay, and they are secreted by, okay, they are secreted by organisms, living cells. Okay, but in this case, as I told you, they are integral part of the cell wall. They are the integral part of the cell wall, okay, and secretion occurs outside the cell, and in this case secretion does not occur until the death of the cell, okay. And for the extraction of these toxins, which we need is cell lysis, but in this case, as exotoxin case, it is not required because these toxins are basically released. Whereas in the case of endotoxins, we need to lyse the cell. We need to lyse the cell in order to isolate the toxins. Okay. Exotoxins are basically heat labile ones. Okay, exotoxins are basically heat labile ones when it comes to stability to the heat, whereas the endotoxins are heat stable, which can withstand up to 250 degrees Celsius also. Okay, and the exotoxins are basically filterable. Why? Because they are very small in size as compared to the endotoxins, whereas endotoxins are not filterable. Why? Because they are the integral part of the cell wall. Okay, by boiling, by boiling it can be denatured. Why? Because of the heat level, because they are heat level. This cannot be denatured because they are not heat level. Okay, in the case of enzymatic activity, mostly have an enzymatic activity, but these endotoxins have a limited enzyme activity, as a limited enzymatic activity. Okay, now comes the specificity. Okay, now comes the specificity. Exotoxins are enzymes basically. This makes them highly specific in their mechanism for their host cells. For their host cells. Okay, so exotoxins, okay, are highly specific. Okay, exotoxins are highly specific. Okay, whereas the endotoxins are comparatively not very specific in nature, are not very specific in nature. Okay, but when it comes to specific receptors, as the exotoxins are highly specific, as the exotoxins are highly specific. Okay, so it will have a specific receptors and usually binds to the specific receptors only, whereas endotoxins, there is no special requirement of the specific receptors. Okay, but there are can be minor variations also, there can be exceptions. Now comes to specificity to the bacterial strain. They are specific even to the certain bacterial strains. Okay, different types of bacteria produce different types of exotoxins, and which may be specific with their function, which may be specific with their functions. Okay, and in this case they are not specific to any bacterial strain. They are not specific to any bacterial strain. Okay, now comes the immunogenicity. They are highly immunogenic. Okay, even a low dose can harm human beings. Okay, even the low dose of these exotoxins can harm human beings. And endotoxins are weakly immunogenic. It requires higher amount of dose. Okay, now comes the fever induction. Okay, it does not generally induce fever. In this case, fever induction by the interleukin 1 production. Okay, toxicity. Okay, as I told you, they are highly immunogenic even in the low quantities. You can see in just the micro quantities it can be fatal, and they are highly toxic. Okay, and endotoxins are basically moderately toxic. Endotoxins are basically moderately toxic. Okay, and fatal in milligram quantity. So they are less toxic as compared to the exotoxins. Okay, now comes the mode of action. Exotoxins have various mode of actions, and mostly by enzymes which are present in the, or the enzymatic activity which is present in the exotoxins. Okay, and endotoxins, it includes interleukins and TNFs. Okay, and potency. Okay, what is the potency of the toxins? In the case of exotoxins it is very high, and in the case of endotoxins it is very low. Okay, in the case of exotoxins, a single toxin molecule can act on a large number of host cells, A single toxin molecule can act on a large number. Okay, even a single ox, as I told you, they are required in very low quantities, whereas endotoxins are required in large quantities to cause a disease. Okay, and what are the harmful effects? Okay, of these toxins? What are the harmful effects? Either they may be cytotoxins, or it may be enterotoxins or neurotoxins with defined action on cells or tissues. With defined action on cells or tissues. Okay, whereas in the case of endotoxins, general symptoms include fever, diarrhea, vomiting, etc. Okay, general symptoms include fever, diarrhea, vomiting, etc. Okay, and exotoxins, as I told you, they are specific. Okay, they may even affect a nervous system also directly. Okay, some may even kill large number of cells in our body also, depending on the type of toxin. Okay, exotoxins, when it comes to neutralization by antibodies. Okay, exotoxins can be neutralized by antibodies, whereas endotoxins cannot be neutralized by the antibodies. Okay, cannot be neutralized by the antibodies. Now comes the detection pattern. Okay, can be detected by many tests. Okay, such as neutralization, precipitation, etc. Okay, you will learn these tests in your semester 3. Okay, of the 603, which includes clinical microbiology. Okay, now comes in the case of exotoxin, which is detected by limulus lysate assay. Okay, which is detected by limulus lysate assay. Okay, and what is limulus? Okay, limulus is just a horse crab. Okay, through which we can detect the endotoxin. Okay, and basically luminous amoebocyte extract is used. Okay, it's an basically aqueous extract of the blood cells, okay, from the limulus crab, which reacts with the endotoxin polysaccharide, okay, which is basically a membrane of gram negative bacteria. Okay, so now moving forward with the lecture. Now next comes the conversion of toxoids. Okay, now comes the conversion of toxoids. In the case of exotoxins, it is possible. In this case, conversion of toxoids are not possible. Okay, what is toxoid first? What are toxoids? Hmm? Toxoids are basically the inactive form of the toxins. As you know, most of the exotoxins are basically proteins. Okay, they lose their toxicity when treated with formaldehyde, although their antigenic properties are retained. So in this form they are called as toxoids, okay, which is an inactive form of the toxin. Okay, and have the ability to stimulate the production of antitoxins, okay, in the body, which is basically they are the antibodies that react with the toxins and utilize them. They react with the toxins and utilize them in the body of the host animal. Okay, why it is important? This is important in the protection of the susceptible host from the disease which are caused by the endotoxins. Okay, for example, toxoids are widely used in as vaccines. Okay, are widely used as vaccines in the case of tetanus and diphtheria. Okay, as well as in the for the treatment of the botulism also, as well as for the treatment of the botulism also. Now comes the availability of vaccines. It is possible. in the form of toxoid in the case of exotoxins, whereas in the endotoxins no effective vaccines are available. Okay, now the disease caused by exotoxins include tetanus, diphtheria, botulism, etc. And endotoxin includes meningococcemia, sepsis by gram-negative rods or various other bacteria, etc. Now examples which produce exotoxins are Staphylococcus aureus, Bacillus cereus, Streptococcus pyogenes, Bacillus anthracis. There are more number of examples. Okay, and now the endotoxins which are produced, okay, are basically E. coli, Salmonella typhi, Shigella, Vibrio cholerae, etc. Okay, this is the different types of uh, sorry, these are the examples of the bacteria which produce exo and endotoxins. Okay, so exotoxins are often divided into various categories. Okay, for instance, okay, please remember these definitions. These are the important terms. For instance, botulinum and texan and the tetanus toxin affect the nerve tissue and are termed neurotoxins. Okay, and the toxins made by Vibrio cholerae which affect the intestinal tract is termed as an enterotoxin. Okay, now comes the acytotoxin, okay, which is basically produced by diphtheria. A diphtheria toxin kills several different types of cell and thus it is termed as acytotoxin. Next comes some cytotoxins may kill leukocytes. Okay, has the ability to kill the leukocytes. Hence they are known as leukocidins. Okay, and some may cause lysis of the blood cells, so they are called hemolysis. So there are a number of different varieties of exotoxins. Okay, and then their names are given according to their function. For example, the exotoxins which affect the nervous system are called neurotoxins. Toxins which affect the intestinal tract are termed as enterotoxin. Toxins which kills different types of cells are termed as acytotoxins, and some cytotoxins, okay, which basically kills cells, and if this case cells are leukocytes, then these types of toxins are known as leukocidins, and some may also cause the lysis of blood cells. It means the toxins which are involved in the lysis of the red blood cells are called as hemolysins. Okay, now there are also some of the role which have been improved in the case of the toxins, okay, such as role of bacteriophages and even plasmids. Okay, as plasmids are the extra chromosomal inheritance which may be present in some of the bacteria and which may also carry the gene for producing the toxins. So how are bacteria, what are the roles of the bacteriophages and plasmids to induce a toxogenicity? So the ability of a bacterium to make a particular endotoxin, okay, basically or the toxogenicity may be due to a chromosomal gene. However, in some instances such as diphtheria or certain types of toxins of the botulism toxin, and the toxogenicity can be conferred on a bacterium as a result of acquiring the temperate bacteriophage that carries a gene for the toxin. Okay, and this process is called as lysogenic conversion. For example, in the case of the diphtheria toxin, okay, its ability is enhanced, okay, due to the presence of the corynephages which undergo the lysogenic cycle, and this gene, okay, this gene basically is carried by the temperate bacteriophage, which are coroniphages, which have affects the Corynebacterium diphtheria. Okay, another one, bacteria can also become toxigenic by acquiring a plasmid that carries a gene for a toxin also. For example, in some strains of E. coli, which are capable to cause diarrhea because they contain plasmid whose DNA encodes for an enterotoxin also. Okay, and another plasmid mediated toxin, which is enterotoxin, is made by food poisoning strains of the Staphylococcus aureus Okay, now let me discuss some of the mechanisms in some of the exotoxins. Okay, now starting with the Clostridial alpha toxin, which is produced by the organism Clostridium perfringens. What is its mode of action, or the action, which type of toxin it may be? It may be hemolysin as well as cytotoxin. The cytotoxin. Okay, in which they have ability to lyse the red blood cells and also kill a variety of other cells also. Okay, and mechanisms. Okay, they produce phospholipase enzyme and hydrolyzes lecithin, thereby damaging the cytoplasmic membranes of blood and tissue cells. Thereby damaging the cytoplasmic membranes of blood and tissue cells. This is how cells are killed. Now comes the staphylococcal toxin, produced by Staphylococcus aureus. It may be hemolysin or leukocidin or cytotoxin. Mechanism is still uncertain, and it may be due to the disruption of the hydrophobic region of the cytoplasmic membranes, or the conversion to an active protease, or due to the conversion of an active protease. Okay, still the mechanism is uncertain, no doubt. Okay, now comes the staphylococcal enterotoxin. The organism which produces is Staphylococcus aureus again. Okay, and the action is of an enterotoxin which causes nausea and vomiting. Okay, mechanism is unknown. Now comes next toxin, Panton valentin factor, which is again produced by Staphylococcus aureus. Okay, action is of a leukocidin. Okay, is of a leukocidin which kills white blood cells or leukocytes. Mechanism causes degranulation of the leukocytes. Okay, next comes, okay, is an LT toxin. It's an LT toxin which is produced by Escherichia coli. Okay, it's an enterotoxin which causes diarrhea, and mechanism similar to that of cholera toxin. Okay, and now next comes the exotoxin A. Okay, organism involved is Pseudomonas aeruginosa. Mode of action is of a cytotoxin, and its mechanism is similar to that of a diphtheria toxin. Okay, now next comes the anthrax toxin, which is produced by Bacillus anthracis, and possibly it's a neurotoxin which affects the nervous systems. Mechanism is unknown, but the studies till now which have been made, the toxin probably acts on the central nervous system, causing respiratory failure. And the toxin consists of three distinct proteins, EF, PG and LF, and no single one of these is lethal, but the combination of all three or just even two can be lethal. Now next comes the pertussis toxin, which is produced by the organism Bordetella pertussis, and what is its action? It's of a cytotoxin. It's of a cytotoxins. Okay, their mechanism is unknown, but causes increased sensitivity to the histamine. Okay, causes increased sensitivity to the histamine and cause an increase in number of leukocytes in the blood and causes hypoglycemia. Okay, what is hypoglycemia? It is subnormal levels of the glucose in the blood. Okay, now comes the erythrogenic toxin. Okay, erythrogenic toxin which is produced by the bacteria Streptococcus pyogenes, which is produced by the toxin Streptococcus pyogenes. What is the basic mechanism of action of an AB exotoxin? Of a AB exotoxin. So the action of AB exotoxin, which I am going to uh, explain, okay, it's just a proposed model for the mechanism of toxin of the action of the diphtheria toxin, of the action of the diphtheria toxin. Okay, so disease caused by bacteria that produce exotoxins often caused by minute amount of exotoxins and not by the bacteria themselves. So it is the exotoxins that basically produce specific signs that produce specific signs and symptoms of a disease, and thus exotoxins are disease specific. Okay, are disease specific. For example, in the case of a botulism, okay, is usually due to the ingestion of exotoxin. Okay, and not the bacterial infection, but not the bacterial infection. So right now I'm going to discuss AB type of exotoxin, but still there are more two other types which are basically membrane disrupting toxins also in this, and some are also termed as super antigens. Okay, some are also termed as super antigens. What are super antigens? Okay, I will explain you later. So right now I'm going to start the process of the mechanism of the AB toxin. Now what happens? A bacterium, okay, a bacterium where exotoxin is produced by the bacteria, where A is the active one portion and B is the binding portion, and B is the binding portion. Okay, so in first scenario, a bacteria produces and releases AB toxin. Bacteria produces and releases a AB toxin. Now comes the binding. Okay, binding to a host cell receptor. Now this is a host cell where this exotoxin binds, where B, which is a binding component, of the exotoxin, attaches to the host cell receptor. Basically attaches to the host cell receptor. Okay, and this function is to bind to the host cell. Now what happens next? Okay, the plasma membrane of the host cell invaginates, which basically you can see folds. Okay, the plasma membrane folds inward. Okay, folds inward at the point where AB toxin and plasma receptor make contact. Okay, plasma receptor make a contact and the exotoxin enters the cell by receptor mediated endocytosis. It's a process of the endocytosis, where a receptor is basically used. In this case, the receptor will be on the plasma membrane. Now, in the next scenario, what will happen? The AB toxin and the receptor, okay, and the receptor are enclosed, okay, in a pinched off portion of the plasma membrane during the pinocytosis. And what is pinocytosis? What is pinocytosis? Okay, anyone? Okay, sorry, I forgot. It's just an fluid endocytosis. It's a mode of endocytosis in which a small particle suspended in the extracellular fluid are brought into the cell through the invagination of the cell membrane. Okay, and this results in the suspension of particle within the small vesicle. Okay, it's called as pinocytosis. Now next one, moving forward, the next part, what happens? AB components of the exotoxins separate and the A component alters the host cell function. Okay, and the A and B will separate, and the A basically will start altering the host cell function, often by inhibiting the protein synthesis, Now the B component at the same time is released from the host cell and the receptor is inserted into plasma membrane for the reuse. Meanwhile, these toxins in which A portion will basically inhibit the protein synthesis. Okay, so toxins, I already discussed the to toxoid point, so I'm just going to move forward with the endotoxins. And what the before moving forward with the endotoxins, let me discuss with you uh, the concept of the superantigens. Okay, superantigens are basically the antigens that provoke a very intense immune systems. Okay, and they are the bacterial proteins through a series of interactions with the various cells of the immune systems. And the superantigens non-specifically stimulate the proliferation of the immune cells called T cells, and these cells, okay, which are basically the white blood cells that can act against foreign organisms. Okay, and also in the case of the foreign, in the case of transplants, and also regulate the activation and the proliferation of the other cells of the immune systems. So in response to superantigens, these T cells are stimulated to release enormous amounts of the chemicals called cytotoxins. Okay, are stimulated to release enormous amounts of the chemicals called cytokines. Okay, cytokines, yes. Cytokines, what are cytokines? They are the small protein molecules produced by the various body cells, especially T cells, that basically regulate the immune responses and mediate cell to cell communications. Okay, and the high levels of cytokines released by T cell enter the bloodstream and give rise to a number of symptoms, okay, which may be fever, nausea, diarrhea, sometimes shock, and in more complicated, it may also lead to death. And bacterial superantigens, of which some of them basically include staphylococcal toxin also, which is basically involved. It's an toxic shock syndrome. As a short, it is also known as toxic shock syndrome due to the higher production of the cytokines. Okay. Now moving forward with the mechanism of the endotoxins. Okay. In the case of mechanism of the endotoxins, as you know, endotoxins are lipid portion of the lipopolysaccharides, and they are present as an integral part of the cell wall of the gram-negative bacteria. Okay, and they are released from bacterial surface by natural lysis of the bacteria and are responsible for various biological effects in the host, of which the first one of which uh, the first one is the macrophage activation. Basically, endotoxin binds to the specific receptors on the macrophages and stimulates die-release of the acute phase cytokines such as interleukin 1, tumor necrosis factor, IL-6 and nitric oxide and the prostaglandins which cause fever, and in inflammation and activation of immune T and B cells. Immune T and B cells. Next comes the complement activation. Next comes the complement activations. Now, the higher concentration of the endotoxin can also activate the alternative pathway of the complement. Okay, basically release of C3A, complement to C3A and C5A to promote inflammatory cells. Okay, chemotaxis, and also may lead to high grade fever, hypotension and shock produced by vasodilation and the capillary leakage. And next one is endothelial activation, which leads to increase in vascular permeability. Okay, it's not permeability, it's permeability. No, it's permeability. And next comes the platelet activation, which basically leads to the mediators that cause increase in vascular permeability, even thrombosis and disseminated intravascular coagulation. Okay, disseminated intravascular coagulation. And what is thrombosis? Thrombosis is a process of a blood clot, also known as thrombus, and this clot can block or obstruct blood flow in the affected area as well as cause serious complications if the clot moves to a crucial part of the circulatory system. Okay. Now next comes the coagulation pathway activation, okay, in which it activates Hagman factor and other coagulation factors, which leads to thrombosis and disseminated intravascular coagulation. Okay. And what is Hagman factor? Hagman factor, okay, is basically a coagulation factor number 12, and it's an plasma protein. Okay, it's an plasma protein and it's an clotting factor. Okay, so the next one is an mast cell activation. It's an mast cell activation which leads to release of mediators, example histamine, that causes muscle contraction and increase in vascular permeability Okay, in gram negative septicemia, endotoxins are released in large quantities causing high fever and petechiae What is petechiae? They are the skin lesions resulting from the dicapillary leakage. Okay, and also the DIC. What is DIC? It says disseminated intravascular coagulation, which may result in shock and possibly death. Okay. Now this is just an example of an endotoxin, and due to which a pyrogenic response occurs. It's a proposed mechanism by which endotoxin cause fever. Okay, where a macrophage which which uh, basically basically ingests a gram-negative bacterium. Okay, which contains a endotoxin. What happens next? The bacterium is degraded in a vacuole. Bacterium will be degraded due to the action of the macrophage. Okay, that induce macrophage to produce cytokines, interleukin 1, as well as tumor necrosis factor alpha. Okay, which releases cytokines and interleukin, and this basically are released in the blood vessels. It pass through the blood vessel, and they signal, and these cytokines are further released in the bloodstream by macrophages, through which they travel to the hypothalamus and the temperature control center of the brain. Okay, which has a prostaglandins, and these cytokines induce hypothalamus to produce prostaglandins. Okay, which reset body's thermostat to a higher temperature, producing the fever. Okay, this is how the antitoxin mechanism works. Just a proposed mechanism. I am again repeating the whole procedure. A macrophage basically ingest a gram-negative bacterium. Macrophage basically ingests a gram-negative bacterium. A bacterium is degraded in a vacuole, releasing the endotoxin that induce the macrophage to produce cytokines, interleukin 1 and tumor necrosis factor alpha. The cytokines are released in the bloodstream by the macrophages, through which they travel to the hypothalamus, the temperature control center of the brain, and the cytokines induce hypothalamus to produce prostaglandins, which reset body's thermostat to a higher temperature, producing the fever. Okay, this is how endotoxins work. Okay, before moving that, I also like to give you an example of one of the streptolysin. Okay, of one of the streptolysin. What is streptolysin? It is basically streptolysin O, which is produced by Streptococcus pyogenes. Okay, and this streptolysin O and streptolysin S is basically inactivated by the oxygen, and it has a multiple activities based on its ability to damage the cell membranes. For example, it is an hemolysin. Okay, and beta hemolysis can be studied around the colonies of the blood agar incubated anaerobically, and sometimes it has also action of the leukocidin. Okay, also has an action of the leukocidin. Okay, so now moving forward with our next topic of today's lecture, which is a very short one, which includes natural resistance, okay, racial, individual, and internal and external defense mechanisms. Okay, now this is just an chart of the defense mechanism, which includes natural and internal defense mechanisms. Now moving forward, you have already studied the internal defense mechanisms, okay, which involves non-specific defense mechanisms and specific acquired immunity. In non-specific defense mechanisms, it includes phagocytosis, natural killer cells, interferons, and complement and properdin. In the case of natural resistance, includes species resistance, racial resistance, individual resistance, and external defense mechanisms, which includes natural barriers and other processes such as coughing, sneezing, perspiring, and related processes. We have already studied external defense mechanisms when we were studying normal flora, okay, through which skin and mucous membranes and various other mechanical barriers act against the disease, and also the coughing and sneezing mechanisms through which bacteria basically, or the viruses or any other pathogens are basically kept out of the, are removed from the body. Okay, now the topic which I am going to cover especially is the defense mechanisms, okay, in regarding to the racial, species, and the individual ones. Okay, so the metabolic and the physiological requirements of a pathogen are important in determining the range of potentially susceptible host. Okay, determining the range of potentially susceptible hosts, and the natural resistant host either fail to provide some essential environment factors required by microorganism for the growth, or have a defense mechanism to resist the infection. So in order to the host general health and the state of nutrition and social as well as economical conditions and other intangible factors such as stress, depression, work, etc. Okay, also play a part in the host defense mechanisms which provide the natural resistance to the host. Okay, so the first one, okay, which involves a species resistance, which involves a basically species resistance. And what does it the species resistance involve? Okay, and this resistance to infection varies with a species of animal or plant, but following it may be confined to humans or animals also. So in general, okay, a species resistance have been physiologically more complex than it appears in the animal kingdom, and have also evolved from the time to time, okay, due to the evolution or passing of genetic characters also from generation to generations. Okay, however, basic physiological characteristics of a species such as normal body temperature can also determine whether a organism, okay, can be pathogenic or not. For this reason, many diseases of mammals do not affect fish or reptiles or various other mammals. Okay, for example, frogs being cold-blooded. Okay, for example, frogs which are cold-blooded are basically resistant to the anthrax. Why? Because anthrax needs the temperature requirement of 37 degrees Celsius. Okay. Conversely, if the body temperature of chicken is lowered from 39 to 37 degrees Celsius, okay, it may be susceptible to the anthrax. Okay, as you know, most of the mammals are resistant to the Mycobacterium avium complex. Okay, which basically affects the birds. Okay, and the type of bacterium which is found in cold-blooded reptiles and do not produce infections in mammals generally. Okay, but it do affects the birds. Okay, so the metabolic, physiological, and the anatomical differences between species affect the ability of a pathogen to cause disease. For example, herbivorous animals are often resistant to the diseases of the carnivores, and a variety of reasons. and also the intestinal anatomy between carnivores, ruminants, as well as between the herbivores as well as the omnivores have different types of systems with regarding the functions of the stomachs, intestinal microbial flora, and also the digestive juices which are produced in the system. Okay, so we can conclude from the characteristics species resistance that the inability, the first one is the inability of an organism to cause a disease, in the resistant species under natural conditions. Okay, the second is the production in the resistant species of a localized or transient infection by experimental inoculation as opposed to the generalized or progressive disease in natural susceptible species. And the third one, the induction of the experimental disease in resistant species only by the massive doses of the microorganisms and often by an unnatural route or under unnatural conditions. So there are various other reasons. Now moving forward with the residual resistance. Okay, moving forward with the residual resistance when it comes to breeds. Various animals as well as plant breeds show marked differences in their resistance to certain infectious diseases. Okay, and a well-known example is often Brahman cattle. Okay, it's a type of cattle which are resistant to protozoan parasite responsible for the tick fever in the other breeds of cattle. And this type of resistance, it's an inherited racial characteristics and is indicated by the fact that a crossbreeding in highly susceptible Texas horn with the Brahman cattle produces resistant variety. So in the case of humans also, there are evidences of the racial resistance which is explained on the basis of selectivity and the survival in most cases. Okay. So for example, the presence of pathogen in isolated races results in the gradual selection for the resistant members as the susceptible members die, often progressive infection. Okay, when the introduction of the new pathogen such as tubercular bacillus by the resistant Europeans to an isolated population such as American Indians resulted in the epidemics which decimated the population. Okay, there are also some of the tropical diseases which have been studied for their resistance. For example, tropical diseases of malaria and yellow fever. Okay, okay, where the resistance is basically present in the African population, natural resistance. Okay, and there are also other factors also, physical factors which plays an important role. Now comes the individual resistance, which is purely on the basis of an individual. Okay, for example, it may lead to its background. Okay, its racial background, the genetic one, or its individual's ability for the adaptation to the environment. Okay, and various factors such as its health condition, etc. Okay, this is how an individual resistance works. Okay, it again can have difference between man, woman, child. Okay, and also in the older ages, or it may be in the child, infant ages, etc. So it may have various different types